The larger one here, with the lacy outlook there, that's a ruffled spreader, whereas the other large guy with a flat surface here, that's studded, and then the smaller one here, that's wrinkly. So I work primarily with the colony morphotype wrinkly, and what we're interested in is discovering the genetic mechanism that turns wrinkly to mucoid. So here you can see wrinkly, it's unsymmetrical, and it's turning into mucoid. This has been seen in cystic fibrosis lungs in both directions, from wrinkly to mucoid and then mucoid to wrinkly. In our lab, we're looking at wrinkly to mucoid. And the main question we're asking is, how many different ways can you transition from wrinkly to mucoid based on evolutionary history? So if your parent genotype of wrinkly is more evolutionarily advanced, is the pathway and path length going to be different than if it's less advanced? A little more background on wrinkly as well as the mucoid type. Wrinkly is associated with the highest level of patient mortality, and mucoids are commonly found near wrinkly in the lung, but little is known about them and their possible deleterious effects. So a similarity that all the parent genotypes wrinkly had, we start with 8, is they all had mutations in the Wisp operon. Now what the Wisp operon does is it's a surface sensing regulator, and it's involved, we don't know the mechanism of how it's activated, but we do know that it's involved with cyclid GMP, which has a direct relationship with biofilm formation. Now we have all this DNA over here, right? And the Wisp operon is only 5 to 10 kilobases, and we have all these various mutations within it. So this is a pretty big deal. Okay. So here's a schematic of the main question again. So we have early mucoid and late. And basically, we grouped the mucoids based on their parent genotype, and the parent genotypes bins are as followed. So you have early, which has 1 Wisp mutation, whereas late has more than 1, but less than 5 mutations. So we're wondering, is this path of early different than this path of late? So at the end of the summer, we sequenced the mucoids that we collected, but first we wanted to look at phenotypes. We had a couple different phenotypes we were really interested in: swarming, or motility, the ability of the bacteria to move around on the surface of the plate, biofilm formation, EPS production, which is a chief component of biofilm, and then biofilm fitness. Based on our binning system of the mucoids versus early and late, we saw stark phenotypic differences. With regard to EPS, early mucoids exhibited almost no EPS production, whereas late mucoids had an awful lot of significantly higher EPS production. Now, biofilm formation of mucoids is generally less than their parent wrinkly genotype, but in the cases where the biofilm formation was greater, it was always a late mucoid that was able to exude that. One of the most interesting phenotypes we found this summer was the swarming/motility phenotype, and here's why. So on the right, this story comes in 2 pieces. On the right, you're going to see what the plate looks like to start with. And you might be able to see 2 concentric rings right in this region here around the colony there. And what that might be is rhamnolipids. And rhamnolipids are a surface surfactant, which means they lower the surface tension and allows the bacteria to more easily spread across the surface of the plate. After 48 hours in an incubator, this is what we find, right? And this only happens to early mucoids. Late mucoids tend to stay this size or smaller. So what happens is they might produce the rhamnolipids, we're not sure, we're still investigating this. And then that allows the bacteria to spread in this unique way. And this is what we call the wild swarming phenotype, and this is the first instance this has been seen in Burkholderia sinus So I talked a little bit about genetics before, and we were able to sequence 27 of 51 representative mucoids. And this is a pretty big deal because 5 to 10 years ago, this would not have been possible, right? And what we found is that there are 2 distinct pathways to become mucoid based on your evolutionary history. So say your parent genotype is early W, right? You have 1 mutation. This is a mutation outside the wisp operon that brings you to the end result of being mucoid. If you're a late wrinkly as the parent genotype, it's very different. You have a reversion mutation within the wisp operon that leads you to the end result. Now we still have a lot of work to do on this project. One of the most exciting parts is comparing our mutations we found in the lab to clinical samples to see how representative they are. We're also going to look at the mutations' influence in different environments, as well as starting new genotypes to see if we can find new distinct pathways. I'd like to say thank you to all the graduate students and undergrads that have helped me in the lab, as well as our funders. And a special thanks to the Hamel Center for Undergraduate Research for funding my freshman REAP, and also Vaughn Cooper for giving me a chance in the lab and all the support he's given me over the last 4 years. Any questions?
You looked at putting artificial or other surfactants in the media and see if that helps them result in the spreading out?
We haven't done that yet, but that's something we're looking forward to doing this summer.
Any other questions?
You said that the wrinkle you're looking at is related with the highest mortality.
Yep.
Is the information you find from this going to be able to relate to the other types, or do those need to be researched separately on their own? Is there enough of a difference between them?
Yeah, there's a pretty big difference between them. So if mucoid types generally mutate only from the wrinkly type, so there's not too much of an overlap there, but they also come in a mixed community. So you'll have the 3 types within the community, so we might do some work with mixed populations as well.
Another question? Alrighty, thank you, Cody.